Hello everyone and welcome to today's live webinar, Exploring Energy Balance Mechanism in Rodents Using Ad Hoc Tools, presented by Dr. Daniela Cota. My name is Diane and I will be moderating today's webinar. The presentation will last approximately 20 minutes and we will keep 10 minutes to answer your questions at the end of the webinar. Should you have any question at any time, please submit them in the question box of the GoToWebinar panel. This session is recorded and you will receive the recording after the webinar, but I strongly encourage you to stay until the end as it is the occasion to have our speaker Daniela Cota answer your questions live. Before starting, I will just introduce Neuroservices Alliance for those who don't know us. We are a consortium of CNS contract research organizations. Our team is specialized in vitro and in vivo electrophysiological and behavioral studies. We deliver endpoints facilitating decision-making to our clients for their CNS drug discovery programs. We value direct and efficient interactions with our clients, and we rely on a team of more than 50 PhD scientists. Now it is time for me to give the presentation to Daniela. Hello, Daniela, and I will let you take the lead from here.
Hello, Diana. Hello, everyone. I am Daniela Cota. I am a research director for the National Research Medical Institute in France. And I lead a team at the Neurocentre Magendie where we work on the neurobiology of obesity. And my laboratory is also part of the Optopath platform. So today I will be giving you a short introduction on energy balance regulation and obesity, as well as a quick overview of tools available and a scientific example of a new mechanism that we have discovered in energy balance regulation. Sorry. So what it is energy balance? Energy balance is a physiological process which is under the control of a number of complex biological responses that it is there to continuously adjust energy intake with energy expenditure. The moment in which this regulation is altered, we are into pathology, and one of these pathologies in which alteration of energy balance are evident is obesity. So why are we interested in studying obesity? We are currently faced with a global obesity epidemic, and unfortunately, the therapeutic options that we have to treat these metabolic disorders are very little. And this is due to a knowledge gap in the underlying biological processes that regulate energy balance. In this context, it's clear that the brain has a critical role. In fact, studies like the one that I'm sharing here with you have, for instance, shown that gene polymorphisms that are associated with an increase in the body mass index are particularly highly expressed in the human brain, and specifically the level of structures such as the hypothalamus or the limbic areas, which are critical for the regulation of food intake and body weight. So then when we look at the panorama of pharmacological options that we have for obesity, it's clear that several of these drugs, some of which actually have been recently also removed from the market, have mostly a central action. And what it is troubling in a way is that for them, for many of those, we don't yet know the exact underlying mechanism of action. So, as I said, the brain plays a critical role in energy balance. And in this context, we are particularly interested in the function of hypothalamic circuits that by their ability to interact with the periphery, thanks to information coming through nutrients, hormonal, and nervous signals, can then modulate food intake and use of energy. Now, one thing that I'd like to point out is that what we eat actually can directly impact the function of these hypothalamic circuits. So much so that food that it is particularly rich in calories, rich in fats and sugars, can rapidly alter the crosstalk between the brain and the periphery, leading to a vicious cycle favoring further intake and the storage of calories, and therefore leading to pathology. Now, in my team, we actually use an holistic approach to look at different parameters that might be relevant for understanding the biology of energy balance regulation. And among the tools that we currently have available, I need to mention some that we're able to acquire and develop through the Optopath platform. So we have specifically developed cages that we call obesity cages that allow us for an in-depth study of feeding behavior in mice. These cages allow to perform operant and Pavlovian conditioning, and they can be coupled with in vivo optogenetics and calcium imaging so that we can couple the image of and the activity of cells, neurons of interest, with the actual analysis of feeding behavior. So while this approach allows us for in-depth analysis of food intake, we can also look at the other arm of energy balance, so energy expenditure. And in particular, we use a system with indirect calorimetry that by allowing recording the use of gases, so the consumption of oxygen and the production of CO2, it allows us to evaluate energy expenditure under basal condition, under exercise, under ambient temperature challenge, and this can be, of course, then associated with analysis of in-cage activity and food and liquid intake. Now, if we come back for a moment to this schema that I've shared with you before about the biological mechanism implicated in the physiopathology of energy balance and the physiopathology of obesity, I wanted to highlight the interest that might be particularly for signals that are coming from the periphery and that might interact with the brain. And a few years ago, actually, our interest went for bile acids. So what are bile acids? Bile acids are lipid derivatives that are synthesized by the liver and that are produced in association with food consumption during the meal because they help digest the fat that it is present in the meal. Now, the interest for bile acids in the metabolic fields have been particularly highlighted because of studies that have been linking the metabolic benefit of bariatric surgery, which currently is the only successful long-term therapeutic option for obesity, with the action of bile acids. Indeed, studies that have been carried out in humans have shown that patients that suffer from obesity have a decreased circulating level of bile acids. However, when these same patients undergo bariatric surgery, these levels immediately increase and stay increased after more than 2 years from the surgery. Other studies have further defined the role of bile acids in the regulation of peripheral metabolism. Indeed, it has been shown that bile acids can not only modulate the digestion and absorption of fat, but within the gastrointestinal tract, they can also modulate the secretion of important hormones such as the GLP-1. They can modulate lipid and glucose metabolism in the liver. They can modulate insulin secretion in the pancreas. They can also affect thermogenesis and energy expenditure in the brown adipose tissue and in the muscle. And finally, they can modulate inflammatory responses through action onto immune cells. So bile acids actually have all these actions by binding and activating specific receptors. Some of these effects are due to their binding to nuclear receptors called FXR, while other metabolically beneficial effects are due to the action of bile acids through a membrane-bound G-protein coupled receptor that it is known with the name of TGR5, which stands for Takeda G-protein receptor type 5. Now, evidence in the literature suggests that pharmacological TGR5 activation in peripheral organs is beneficial for weight loss and for type 2 diabetes. So we actually knew that in the periphery, we have expression of TGF5 receptors. The question was whether these receptors are also present in the brain and whether bile acids might actually directly act within the brain. So again, the literature came to our help because there was already evidence suggesting expression of the TGF5 receptor within the brain and particularly within the hypothalamus, both in rodents and in human tissue. So we basically wondered whether there is this system at the level of the hypothalamus might be altered by obesity and whether it might then have a role in the physiopathology of obesity. So to answer these questions, a great part of the work was actually carried out by 3 people in my laboratory. And first, we evaluated the possible changes in bile acids and related components of the bile acid, the TGF5 system within the hypothalamus. First, we observed that diet-induced obese animals actually show a decrease in the levels of bile acid within the hypothalamus, which is accompanied to a decrease in circulating levels of bile acids, exactly like I previously showed you in patients suffering from obesity. Moreover, when we started looking at different bile acid species, we actually found that a bile acid that goes with the name of deoxycholic acid, so DCA, which is a very strong and potent TGF5 agonist, is particularly decreased in the hypothalamus as well as in the plasma of diet-induced obese animals. We then looked at some changes in the molecular expression of bile acid transporters and of the TGF5 receptor. As I told you, bile acids and their levels in the circulation increase actually in relation with the meal. So what you see here is a comparison in lean animals this time under fasting condition and after a meal, so animals that were actually fed. And this nutritional challenge led to an increase in the expression of bile acid transporters, which are the 3 molecular markers that you see here within the hypothalamus, which was accompanied by a decrease in the expression of the TGF5 receptor. So this basically suggests that there is a dynamic modification of the molecular machinery that allows bile acids to enter into the hypothalamus and then have biological effects. However, when we repeated these same experiments in obese animals, this time we were not able to see any difference in terms of the expression of these markers in response to food consumption. Then, having found that there is an alteration of the system within the hypothalamus, we used pharmacology, and we particularly picked a very selective TGF5 agonist, so this was a synthetic agonist, to assess whether its central administration, which was done by intracerebroventricular injection, could affect food intake and body weight in obese animals. So here the data, you clearly see that the compound dose-dependently decreased 24 hours food intake and body weight. And moreover, we actually found similar effects on food intake and body weight also when the compound was directly administered within the mediobasal hypothalamus. So having found that there is an acute action of the pharmacological activation of TGF5 on body weight and food intake in obese animals, we went on and treated the animals chronically with the TGF5 agonist. And chronic treatment over 4 weeks was able to decrease body weight in obese animals. This effect was associated with a decrease in fat mass without changes in lean mass, and also to a decrease in the cumulative food intake over the period of the treatment. We then evaluated the feed efficiency, which is the ratio between the body weight gain and the calories ingested for the period of the study, so 4 weeks. And as you can observe, the feed efficiency was actually negative for the animals that had been receiving centrally the TGR5 agonist. This basically suggests that a mechanism other than a decrease in food intake might have been participating to the decrease in body weight. And one possible mechanism that might be implicated is, in this case, an increase in energy expenditure. So to evaluate energy expenditure, we put our animals in our indirect calorimetry cages. And this experiment actually was done before any differences in body weight were actually evident. And you can clearly see that the treatment with the TGR5 agonist clearly increased energy expenditure, particularly during the dark phase. So this increase in energy expenditure suggests that there is an involvement possibly of the sympathetic nervous system that induces this increase. And to evaluate whether this was the case, we therefore repeated this same analysis in the same animals, but this time in thermoneutrality, a situation in which the sympathetic nervous system is not anymore recruited. And as you can see, under this temperature modification, we could not observe anymore the differences between the vehicle and the treated group. So to further then evaluate whether the sympathetic nervous system is relevant for the ability of central TGR5 activation to modulate body weight and fat mass, we decided to combine the central pharmacological administration with peripheral chemical sympathectomy. So here our animals were pre-treated with 6-hydroxydopamine to induce peripheral sympathectomy, and then were receiving again chronically the central TGR5 agonist or its vehicle. And as you can see from the data, this time the chemical sympathectomy strongly blunted the ability of the TGR5 agonist to modulate body weight. And we could also not anymore observe the differences that I previously showed in terms of changes in fat mass and in food intake. So having found that stimulation of the TGR5 within the brain is relevant as a mechanism that can counteract obesity, we wondered whether the physiological activity of TGR5 is actually relevant in the context of the regulation of energy balance. And to answer this question, we actually used genetic tools that have allowed us to downregulate the expression of TGR5 specifically within the mediobasal hypothalamus. So here we had basically used, thanks to one of our collaborators, TGR5 flux animals, that received an injection of a viral vector expressing either the CRE or the control protein GFP within the mediobasal hypothalamus. And as you can see, this treatment led to a 50% decrease in the expression of TGR5 within the mediobasal hypothalamus. What happens in terms of the phenotype of these animals? So as you can see here, we had lean TGR5 flux that underwent treatment with a control viral vector or the CRE. And we also had a third group in this specific experiment with black 6 mice that received the AAV CRE just to control for possible off-target effects. So as you can see, once that we put the animals onto high-fat diet, the only group that actually gained weight in response to the high-fat diet much more than the others was the group that had been knocked down, had knocked down in the expression of TGR5 in the mediobasal hypothalamus. This was associated with an increase in fat mass, with an increase in food intake, and an increase in feed efficiency. When then we repeated the same experiments, but this time we used already obese TGR5 flux animals that underwent the exact same genetic manipulation. Again, we found that the knockdown of TGR5 within a mediobasal hypothalamus further increased body weight, further increased fat mass, cumulative food intake, and feed efficiency, overall suggesting that the TGR5 within the mediobasal hypothalamus is actually protecting from obesity. So is in this condition the sympathetic nervous system relevant? Again, we placed our animals in our indirect calorimetry cages, and this time we repeated the same genetic manipulation, but under thermoneutrality. And under thermoneutrality, we could not see any more differences between our control and pre-treated TGF-5 flux animals. To confirm that knockdown of TGF-5 within the hypothalamus results into an alteration of the sympathetic nervous system, we then challenged the animals with cold, because cold is known to induce a recruitment and activation of the sympathetic nervous system. And this usually leads to an increase in the expression of thermogenic markers such as UCP1 in the brown adipose tissue or phosphorylation of the hormone-sensitive lipase within the white adipose tissue. And as you can see, these two molecular markers, the expression of these two molecular markers was actually blunted in our animals that had undergone downregulation of TGF-5 in the mediobasal hypothalamus. So in conclusion, the data that I have shown you illustrate a new mechanism that we have discovered in the context of diet-induced obesity, and this new mechanism is given by the bile acid TGF-5 signaling within the hypothalamus. Our data suggest that pharmacological stimulation of central TGF-5 might be tackling obesity by stimulating the sympathetic nervous system. Vice versa, the genetic suppression of TGF-5 favor further obesity by downregulating the activity of the sympathetic nervous system. So opposite modulation of the sympathetic nervous system then has an opposite impact on body weight, food intake, fat mass, and energy expenditure. So I'd like to conclude here. Of course, I'd like to thank all the members of my laboratory and the specific collaborators that were part of the study that I have just shared with you, as well as our funding. And are there any questions, Diane?
Well, thank you very much, Daniela. We have a couple questions, so I will start right away. And for the attendees, of course, please feel free to ask additional questions now. So could you give us some details on the type of neurons that may be involved in mediating the effects that you describe?
Yes, of course. So this is something that I did not share in the presentation, but we have been actually looking at what were the possible neurons that might be expressing TGF-5. And this was done by using FACS coupled with quantitative PCR, and we found that the different type of neurons within the hypothalamus actually express TGF-5, both GABAergic and glutamatergic neurons, as well as the neuronal populations that are known to modulate food intake and body weight, particularly located in the arcuate nucleus of the hypothalamus, like HRP neurons and POMC neurons.
Okay, thank you. Is the TGF-5 receptor expressed in brain cells other than neurons, and what's its role in there?
Yes, of course. So TGF-5 is not only expressed by neurons. There are studies that have shown that TGF-5 is also expressed by non-neuronal cells, so by glia, by astrocytes, and by microglia. It is not known yet whether there might be a role, for instance, in the context of neuroinflammation. We did do specific experiments to rule out that the effects that we were observing were not due to astrocytes, for instance. In fact, the viral approach that I have described in the last part of my presentation was used to target the expression of TGF-5 in astrocytes. So in that case, we used a viral vector, whose expression was driven by the GFAP. And in this case, we could not see any differences after the deletion of TGF5 in GFAP positive cells in terms of changes of body weight, fat mass, or food intake. So this overall, together with our analysis of the expression of TGF5 and the viral approaches that we use, which was targeting neurons, suggests that the metabolic effects that we observe are mainly driven by neurons.
Okay, thank you very much. I have another question. Do you have evaluated any drug in your model?
Apart from evaluating the effect of the TGF5 agonist, no, we haven't.
Okay, and also now, to what human metabolic disease is your model most relevant?
But definitely for obesity as well as for type 2 diabetes. So the animals that we use for all our studies, both for the pharmacology of the TGF5 receptor as well as the genetic approach, were animals that were kept on a high-fat diet for a number of weeks. They were on a high-fat diet for at least 3 months, and they clearly also had insulin resistance. We did not look into the effects onto insulin resistance, but probably there might be some.
Okay, thank you. Now we have a final question. What confidence do you have in the translatability of your rodent model to human metabolic imbalance?
Well, actually, there are a number of studies that have shown the translatability of the use of animal models like the ones that we are using towards humans. There are a number of published papers that have clearly shown that.
Okay, thank you very much. We do not have any additional question for the moment. So do you wish to add anything in particular, Daniela, before we conclude?
No, I hope that the people that have attended have found the presentation of interest, and maybe this might spark further discussion.
Yes, for sure. So, well, thank you very much. And we will follow up with an email with the opportunity for you to ask additional questions and get in touch if you want. Thank you very much for attending today. Thank you, Daniela.
Thank you.
And wish everyone a good day.